Module 16, Chapter 23, Part 2. Now that you've become familiar with the process of glomerular filtration, let's talk about what happens in the renal tubule. One thing that happens there is tubular reabsorption. Tubular reabsorption is the process of reclaiming water and solutes from your tubular fluid and returning them to your blood. Tubular reabsorption occurs through two routes. One route is called the transcellular route. In transcellular reabsorption, you are going to have your solutes and water passing across the apical surface of the cell, passing through the cytoplasm of the cell, and then out of the cell along its basal surface into your kidney interstitial fluid. From the kidney interstitial fluid, these substances can then move into either your peritubular capillaries or the vasa recta. Now, this particular type of reabsorption is a selective process because moving into the cell across the plasma membrane or moving out of the cell across the plasma membrane requires the use of transport proteins. The other way to reabsorb is through a paracellular route. In the paracellular route, substances are going to pass through the gaps between the cells. Now, this type of movement is referred to as solvent drag because for the most part, as water moves in between the cells, it just drags other solutes along with it. The other process that occurs in the renal tubule is tubular secretion. Tubular secretion is a process in which the renal tubule extracts chemicals from your blood and secretes them into your tubular fluid. This is also a selective process because it's going to require you to use membrane proteins to occur. Now, most reabsorption and secretion are subjected to a transport mechanism. The transport maximum is a maximum rate for the movement of a substance. And the transport maximum exists because there is a limited number of carrier or transport proteins for a particular substance. Your transport maximum is going to determine your renal threshold. The renal threshold is the plasma concentration in which a specific compound or ion begins to appear in your urine. For example, glycosuria is when you begin to see glucose in your urine. Now, glycosuria will occur if your glucose concentration in your plasma exceeds 180 milligrams per deciliter. Aminoaciduria is when you actually start to see amino acids in your urine, and this occurs if your amino acid levels exceed 65 milligrams per deciliter. Now, water-soluble vitamins have very, very low renal thresholds. So this is why when you're taking water-soluble vitamins like vitamin C, it's best to take vitamin C in very low doses at periodic intervals across a day versus a huge megadose at one time. You can take that big 1,000 milligrams of vitamin C, that big tablet, you feel a cold coming on, you take that big dose of vitamin C. However, all of that vitamin C is going to enter your blood. And when that high concentration passes through the kidney, all of the vitamin C is going to get filtered out at the renal corpuscle. Your renal tubule will only be able to reabsorb just a really small fraction of it. So in essence, you spend all of this money on this big bottle of expensive 1,000 milligram tablets of vitamin C, and pardon the phrasing, but you do end up pissing your money away. Because most of that vitamin C is actually going to be in your urine. because your kidneys they just cannot reabsorb that much. So what they actually recommend is to get the 200 milligram tablets and take those over, spread them out in 5 doses throughout the day. You'll lose less in your urine, and you'll actually maintain a more steady level of vitamin C within your plasma throughout the entire day versus that single megadose. So just be careful with the water-soluble vitamins. It's always better to go with a lower dosage and space them out throughout the day because of this really low renal threshold. Before talking about tubular reabsorption and secretion in the PCT, I do want to mention that the upcoming segment, what's happening in the proximal convoluted tubule, what happens in the distal convoluted tubule, what happens in the collecting duct, from 2-semester book to 2-semester book, these are the areas that have the greatest disparity with regards to content. I actually went through 5 different 2-semester lecture textbooks, all from different publishers, and they did not agree 100% on everything in these particular sections. So what I'm going to do is I'm going to follow what Saladin has. What I actually think is I don't think everyone exactly knows everything that is happening here, and that it depends on what primary literature the different authors read as they put together these particular chapters. So we're going to follow the Saladin description of what's moving and where. Just know if you go to another 2-semester book, there might be some slight differences in some of the details. So I just wanted to put that out there because I know you guys have different backgrounds, you might use different reference texts for extra reinforcement, and I just wanted to put that out there. As an instructor, it makes things very frustrating because there is no continuity from book to book, 100% continuity with this particular set of content. That being said, let's talk about your PCT and what it can reabsorb and what it can secrete. First thing you should make sure you get into that long-term retention is your proximal convoluted tubule actually reabsorbs the greatest percentage of your glomerular filtrate. It reabsorbs 65% of what was filtered in the renal corpuscle. Now, if you start looking at what it reabsorbs and getting a little bit more specific as to the different substances, with regards to water, it reabsorbs 2/3 of the filtered water, meaning 60 to 70% of what you filtered in the renal corpuscle is going to be completely reabsorbed here. Now, this water movement is through osmosis. So water is following its osmotic gradient from the lumen of the proximal convoluted tubule back into your blood in the peritubular capillaries. The water movement can be paracellular, so it could just be moving between the cells. It could be transcellular through aquaporins, which are water-specific channel proteins. This water movement is usually referred to as obligatory water reabsorption because it is occurring by osmosis and it can't be prevented. So this is, I like to think of it as the water is obliged or obligated to follow the sodium, to follow those solutes. With regards to electrolytes, this could be a full module unto itself if you get into all of the different electrolytes that are filtered in the renal corpuscle. We're just going to hit some of the big ones. For example, sodium. Sodium reabsorption is actually key for everything else that is going to be reabsorbed in the PCT. It's creating an osmotic gradient and an electrical gradient that helps with water reabsorption and other solute reabsorption. With regards to percentage, on average you reabsorb about 60 to 70% of your filtered sodium here. which makes sense if you think about the water percentage. Because if you think of water likes to follow salt, if you reabsorb 60 to 70% of the sodium, it makes sense that you would then reabsorb 60 to 70% of the water. With regards to the movement of sodium, there are various symports and antiports in the plasma membranes that are going to help you with sodium reabsorption. Remember from AMP1 that a symport is moving sodium and another substance at the same time and in the same direction. So reabsorbing sodium with a symport could be helping you also reabsorb glucose or an amino acid or lactate. In fact, with regards to your organic nutrients, many move through this co-transport mechanism with sodium. You also have antiports, which are moving two things at the same time, but they're moving them in opposite directions. Specifically, there is a sodium-hydrogen antiport, which is going to help you reabsorb sodium, but at the exact same time secrete hydrogen. So it's accomplishing two different functions: reabsorption of sodium, reabsorption or secretion of hydrogen. Now, earlier I mentioned that angiotensin II will affect your proximal convoluted tubule and cause it to increase sodium reabsorption. This is actually relevant here because angiotensin II activates the sodium-hydrogen antiport. So angiotensin II is going to activate these little antiports, increasing your sodium reabsorption, but at the exact same time, increasing your hydrogen ion secretion as well. So not only is it affecting sodium reabsorption, but it could potentially have an effect on pH as well. Now, sodium ions are positively charged, and your chloride ions are negatively charged. So chloride ions actually follow sodium because they're electrically attracted to it. So you could just have diffusion of chloride following an electrical gradient. There are also various antiports that can help with the reabsorption of chloride. Both sodium and chloride can pass through the paracellular route as well, so they can just get dragged with that during that solvent drag between the epithelial cells, so they can get moved that way too. Potassium, magnesium, and phosphate can pass through solvent drag through the paracellular route. Also, phosphate can be co-transported, so it can go through the symports with sodium and get reabsorbed that way as well. With regards to calcium, you're going to actually reabsorb 66% of your calcium through the proximal convoluted tubule, and this reabsorption is independent of hormonal influence. Now 52% of the calcium specifically is reabsorbed through the paracellular route, and 14% is going to be moved by the transcellular route. With regards to your organic nutrients, they are all reabsorbed here. So when your tubular fluid leaves the PCT, it does not have organic nutrients remaining, unless of course you went above the renal threshold, but we're not going to murky the waters there. With regards to the nutrients, again, as I mentioned before, many move in co-transport mechanisms with sodium. Amino acids move through sodium co-transport, lactate. In fact, there are specific sodium glucose transporters that are going to reabsorb both sodium and glucose together. With regards to nitrogenous wastes, for the urea usually passes through the epithelium with water. The nephron as a whole So not just specifically the PCT. The nephron as a whole does reabsorb 40 to 60% of the filtered urea. So you don't excrete 100% of your filtered urea. Your body does retain about 40 to 60. Your PCT helps with this reabsorption. But just know that the renal tubule as a whole is going to help with the reabsorption of urea. With regards to uric acid, the PCT reabsorbs nearly all of the uric acid entering it, but later components in the nephron, including later parts of the PCT, you do actually secrete all of the uric acid back into the tubular fluid. So all of the uric acid eventually is going to be removed in your urine. Creatinine is not reabsorbed at all anywhere. So all the creatinine that is filtered stays in the the tubular fluid, lost with urine. Also, any lipid-soluble solutes that are in the tubular fluid, they can be reabsorbed by simple diffusion just across plasma membranes into your blood. So this would be fatty acids, or even your lipid-soluble vitamins that do happen to get filtered out. Oh, in the organic nutrients do include your water-soluble vitamins. Right? It does include those. With regards to tubular secretion, your proximal convoluted tubule is the main site for tubular secretion, though there are some of the other components of the renal tubule that do some secretion as part of their function. In the proximal convoluted tubule, and to a much smaller degree, the nephron loop, tubular secretion is going to serve three major purposes. You are going to secrete varying proportions of hydrogen to bicarbonate ions, which are going to help maintain your acid-base balance. You can secrete various wastes, including urea, uric acid, bile salts, ammonia, any creatinine that might still be within the blood, you can secrete it as well. And please remember, the uric acid secretion compensates for its reabsorption earlier in the PCT. And the secretion of uric acid accounts for all of the uric acid in your urine. The tubular secretion in the PCT also helps to clear drugs and contaminants from your blood, such as morphine, penicillin, aspirin, and this is a main reason for why so many drugs have to be taken 3 or 4 times a day, because you have to keep up with this secretion in the PCT to maintain therapeutic levels of the drugs within the blood. Alright, so now that we've gone through the PCT, we're going to look at what happens in your nephron loop with regards to reabsorption. Now, it's important to know that when your tubular fluid is entering the nephron loop, that you remember your nutrients are absent. Those organic nutrients were all reabsorbed in the PCT, and the osmolarity of your tubular fluid is very similar to your blood. Now, the nephron loop poses an issue for instructors when they start thinking about how to try to teach it, because its functions depend on positive feedback, meaning the function of one limb actually allows the other limb to function. So if you have something that's running on a loop, there really is no start and no finish. So the functions of the ascending limb allow the descending limb to function, and the functions of the descending limb allow the ascending limb to function. So I prefer to start with the ascending limb and then tie in the descending limb functions and to show how the nephron loop works. Overall, your nephron loop is responsible for reabsorbing about another 25% of your filtered sodium, potassium, and chloride. However, this potassium that is reabsorbed is going to be secreted back almost immediately into your tubular fluid as well. It also reabsorbs another 15% of your filtered water, and this water movement is also obligatory water reabsorption. So it's water moving along its osmotic gradient, so water being obliged to follow the solute. Alright, so let's talk about your ascending limb. Now, the ascending limb is impermeable to water and most solutes. Now, remember from A&P1, Chapter 3, impermeable means nothing passes. So it's kind of like jail lockdown. So water cannot pass across this wall, and most of your solutes can't. However, the ascending limb can reabsorb sodium, potassium, and chloride, but again, all three get reabsorbed into the kidney interstitial fluid, but potassium is going to be almost immediately then secreted back into your tubular fluid. Now, this reabsorption is active transport. So we are actively reabsorbing, using ATP, these three particular ions. And what this is going to do is it's going to decrease the amount of solutes that are in your tubular fluid, and at the same time increase the amount of solutes within the interstitial fluid of the kidney and then also in the blood. And what this is going to do most importantly in the interstitial fluid is it's going to create a concentration gradient in your renal medulla. The highest concentration is going to be very deep in the medulla. The lowest concentration is going to be very superficial in the renal medulla. So we have basically a really, really salty renal medulla. So an environment surrounding this tubule that has a lot of salt in it. Urea also contributes to this concentration gradient. The urea actually comes from urea reabsorption in the collecting duct. Okay, so the collecting duct reabsorbs some urea, and it's also kind of sitting out there in the deep parts of your medulla, adding to this concentration gradient. Now, alright, so now as your ascending limb is actively reabsorbing the sodium, the chloride, the potassium, and secreting the potassium back into the lumen, establishing this really, really salty concentration gradient in the kidney interstitial fluid, your descending limb is doing something completely different. Your descending limb is very permeable to water. So water can cross its walls very easily, but it is completely impermeable to most solutes, especially ions. So there's no movement of sodium, no movement of chloride, no movement of potassium, nothing. So very permeable to water, no permeability to anything else. Before going forward, and before I forget, your descending limb does do some secretion of urea into the lumen of the descending limb. So you can pick up a little bit of urea from your kidney interstitial fluid, well, pull it out of your blood from your kidney interstitial fluid and into the descending limb. What we're going to do is we're going to focus in on what's happening with that water. So now imagine you've got tubular fluid moving from the PCT into your descending limb. And that tubular fluid has a really high concentration of water. It's got a lower concentration of solute. it starts heading in down through the descending limb, and it's passing through a much saltier exterior. Now, we've talked about water concentration before, and if you have lots and lots and lots of solute, you have less water in that area. So if you have very little solute, you have a lot of water. So I want you to imagine we have got your tubular fluid heading down through your descending limb with a low solute, high water concentration, and it's passing through an environment that has high solute, low water. And what you've learned about water is water likes to move from an area of high concentration to an area of low concentration. So because of the concentration gradient created by the ascending limb, you're going to have water moving by osmosis from the inside of the descending limb out into the interstitial fluid of the kidney, and from there back into your blood. So your descending limb reabsorbs water by having it move along its osmotic gradient from the high on the inside of the limb to the low on the outside of the limb. So the ascending limb function allows the descending limb to do its job, which is reabsorb water. Now, you also have to imagine what's happening to the tubular fluid as it is moving down that descending limb. As the water is continuously pulled out of that descending limb, you are increasing the solute concentration of that tubular fluid. By the time you get to the turn in the loop, your tubular fluid is now very low in water, very high in solute. Okay? Very, very low in water, very high in solute, meaning it's got really high ion concentrations. Now, this change in the composition of your tubular fluid is actually important because that high solute, low water concentration actually increases your ion reabsorption in the ascending limb. That high ion concentration increases your rate of active transport of the ions out of your ascending limb. So your descending limb, reabsorbing all that water, creates a nice salty tubular fluid that's going to increase your active reabsorption in the ascending limb. So the function of the descending limb is essential for the ascending limb to do its job as well. The distal convoluted tubule and the collecting duct, their functions are more variable. They both reabsorb variable amounts of water and salts, and that variability is because they're regulated by hormones. So what these two areas do with regards to reabsorption is going to be regulated by hormones. Now, in both of these structures, you're going to find two types of cells. You'll find principal cells, which have receptors for hormones. These hormones are going to then impact sodium chloride and water balance primarily. You also have intercalated cells, which help with the reabsorption of potassium and the secretion of hydrogen ions, and the secretion of hydrogen ions is going to be really important for maintaining your acid-base balance. So let's talk about how reabsorption in the DCT and the collecting duct are regulated hormonally. The good thing is that you've already heard of these hormones before. What we're going to be doing is kind of tying in exactly where they're acting to create their effects. Aldosterone is going to act on the thick segment of the ascending limb of your nephron loop, the DCT, and the cortical part of your collecting duct. And you've already learned that aldosterone is going to increase sodium reabsorption and at the same time increase the secretion of potassium. So we're going to take sodium from your tubular fluid, put it back in the blood, and we're going to get potassium from your blood and put it into your tubular fluid. The sodium effect is very significant because water is going to follow that lovely osmotic gradient, and it's going to want to follow that sodium back into the blood. Also, it's very important for chloride reabsorption because that negative chloride is also going to want to follow that sodium as well. Your atrial natriuretic peptide, your ANP, we mentioned before that it dilates the afferent glomerular arteriole and constricts the efferent glomerular arteriole. We also mentioned that it inhibits things like renin, aldosterone, and ADH. With regards to the structures, it inhibits sodium and chloride reabsorption in the collecting duct. So it's going to inhibit sodium reabsorption in the collecting duct, and then if the positive ions are staying in the tubular fluid, the negative chloride is going to want to stay as well. So by inhibiting the sodium reabsorption, you also end up inhibiting the chloride reabsorption as well. Antidiuretic hormone, ADH. This particular hormone increases the numbers of aquaporins in your collecting duct. So it's going to cause the cells of your collecting duct to produce more of those water channels. And if you have more aquaporins, you can reabsorb more water. So for example, if you are dehydrated, when you're dehydrated, you have higher levels of antidiuretic hormone. If you increase your antidiuretic hormone, you increase the number of aquaporins within the collecting duct, allowing you to increase your water reabsorption. The increase in water reabsorption would help to increase your blood volume, specifically increase that plasma concentration, which would help with the dehydration because you're conserving the water. However, by reabsorbing more water, you're going to decrease your urine volume. So you're going to have less urine volume, and the urine you have is going to be much more concentrated, meaning your urine's going to have more solute, less water. If you are well hydrated, you have very low levels of antidiuretic hormone. The drop in antidiuretic hormone actually causes the aquaporins to be removed from the plasma membranes, meaning the collecting duct does not reabsorb the water, meaning the water remains in the tubular fluid, creating a larger volume of urine, but also more dilute urine. So the urine's going to be high water, very low solute. Now, this particular water movement is referred to as facultative water reabsorption because you're using these aquaporins to facilitate the movement of water across the wall of the collecting duct. It's not water just following its osmotic gradient. You're facilitating the movement. You're actually on purpose increasing or decreasing the amount of water that you're moving. Parathyroid hormone. When parathyroid hormone acts on the proximal convoluted tubule, it actually inhibits phosphate reabsorption. So it actually prevents the PCT from reabsorbing phosphate. So the phosphate stays in the tubular fluid. Your urine will have higher phosphate levels. What this also means is that you're going to have a decrease in your blood phosphate. Now, this is actually important because if your blood phosphate levels are low, it decreases the chances of forming your hydroxyapatite crystals in your bones. You guys learned in A&P1 that hydroxyapatite crystals that are forming your bone matrix, you need calcium and phosphate to form them. So if you don't have the phosphate in your blood, you can't form those crystals. So you're going to end up decreasing bone deposition, which means you're going to be increasing, you're going to keep the calcium in your blood. If you're not depositing bone matrix, your bones are not going to be absorbing calcium from your blood, so it actually helps to keep the calcium in your blood. When PTH acts on your distal convoluted tubule and the thick segment of your ascending limb of the nephron loop, it actually causes these two areas to increase calcium reabsorption. So it's going to help to increase your blood calcium level. Now, the DCT itself is responsible for reabsorbing another 8% of your filtered calcium. The thick segment of the ascending limb reabsorbs about 25% of your filtered calcium. Also, PTH is going to stimulate the kidney to promote secretion of calcitriol. Specifically, PTH is going to tell your proximal convoluted tubule to secrete calcitriol. So the PCT is responsible for producing your active vitamin D, which is then going to go to your digestive tract and increase calcium absorption through your small intestine. Now, just as kind of little side notes, your collecting duct also reabsorbs urea deep in the medulla, and I mentioned this particular function when we were talking about the formation of the concentration gradient by your ascending limb of the nephron loop. I had mentioned that some of that concentration gradient is created by the urea reabsorbed by the collecting duct. With regards to secretion, your DCT does have the ability to do some secretion. It's involved with the secretion of drugs, waste, other solutes that you have in your blood that you still need to get rid of. So the DCT is kind of viewed as the last structure, really tweaking the final solute composition of your urine. Now, let's talk a little bit about water loss. We've talked about we've talked about water reabsorption as we have gone through the renal tubule. So I just kind of want to summarize it a little bit. Water loss from the body through the urinary system is going to be controlled through changes in water reabsorption. Your proximal convoluted tubule and your descending limb reabsorb water through osmosis. And again, this use of osmosis to reabsorb water is referred to as obligatory water reabsorption. It's water movement that cannot be prevented. It's water going from an area of high water, low solute, towards an area of high solute, low water. And overall, the obligatory water reabsorption recovers or reabsorbs 85% of the filtered water. Now remember, the proximal convoluted tubule itself is reabsorbing 60 to 70%, so it's still the PCT is doing most of this work. Now, the DCT and the collecting duct really are very impermeable to water. The DCT does not reabsorb any water, which is why water reabsorption was not even referenced when I talked about the DCT. The collecting duct is impermeable to water. However, it can undergo facultative water reabsorption, so water movement that is more controlled, and that this water reabsorption is dependent on antidiuretic hormone. Remember, antidiuretic hormone inserts aquaporins into the collecting duct, which will enhance the rate of osmotic water movement. So if you have high ADH, you have high levels of aquaporins, you increase your rate of water reabsorption. If you have low levels of antidiuretic hormone, you have low numbers of aquaporins, pretty much no aquaporins, and the collecting duct is relatively impermeable to water. Now, I just want you guys to go back to chapter 18 chapter 17, because we talked about diabetes insipidus when we talked about antidiuretic hormone, and that diabetes insipidus was caused either by a lack of antidiuretic hormone or an inability of the targets, meaning your collecting duct, to respond to ADH. And that in diabetes insipidus, the danger is that you're unable to perform this facultative water reabsorption. You start having large amounts, up to 20 liters per day, of this very watery urine, and you can die from dehydration very quickly. Now, you might have heard of diuretics before. Diuretics are designed to slow your renal reabsorption of water. Okay, so they're going to slow down that renal reabsorption. What this will do is it will increase your urine volume. So it's going to increase your urine flow rate. You're going to be producing urine at greater volumes. But as you have greater urine volume, you will have a decrease in blood volume as well. And that decrease in blood volume will decrease your systemic blood pressure. Now, there are some natural diuretics, so things that you can ingest that just have this particular effect. For example, caffeine. Caffeine specifically blocks sodium reabsorption. And if you don't reabsorb the sodium, you won't reabsorb the same amount of water. The water stays in the renal tubule, increase in urine volume. Alcohol is actually also a natural diuretic because it decreases the amount of ADH in your blood because it blocks its release. And losing the ADH decreases the numbers of aquaporins, and you no longer reabsorb water through the collecting duct. However, many of your drugs for hypertension, remember hypertension is high blood pressure, and many drugs for hypertension are diuretics. For example, Lasix. Lasix is a very popular antihypertensive. Lasix actually targets the ascending limb, and it decreases the sodium reabsorption. And by decreasing sodium reabsorption in the ascending limb, you make the concentration gradient within the renal medulla weaker. And by decreasing that concentration gradient, you actually will then decrease water reabsorption in the descending limb of the nephron loop. And by decreasing the water reabsorption there, you will end up having a greater urine volume. So what happens to your urine once it actually gets into the renal pelvis? And the answer is not much, because when your urine leaves your papillary ducts, there are no other urinary system structures that will alter the composition of your urine. So once you leave the renal papilla, the urine is done. All of your other structures are just performing transportation of that urine to the outside world. So from the renal pelvis, your urine's going to move into your ureters. The ureters are going to move urine through peristalsis. Also, gravity will help, because if you think about position, the kidneys are superior to the bladder, and gravity is going to want to pull that urine inferiorly through your ureters. You also have hydrostatic pressure. You're constantly producing urine at the kidney, and that urine is going to help to continually push fluid out the renal pelvis, down the ureter, eventually landing in your urinary bladder. The urinary bladder is just for urine storage. Its average capacity is about 700 to 800 milliliters of urine. However, it is rare that you will actually fill your bladder to that capacity, because the urge to urinate actually begins at about 200 milliliters. Now, what will happen is, as you are moving urine down into your bladder is going to start to stretch. And in the walls of the bladder, you have stretch receptors. And when these stretch receptors become activated, you begin what is called the micturition reflex. Micturition is the fancy biological word for urination. Now, these stretch receptors are going to send signals up to the sacral region of your spinal cord. And in the sacral region of the spinal cord, there is a center called the micturition center. And this micturition center, when it is stimulated, it's going to send parasympathetic impulses down to your bladder. And what this will do is it's going to cause contraction of the detrusor, which is the muscle within the bladder wall. Contraction of the detrusor increases fluid pressure within the bladder. It also causes the internal urethral sphincter to relax and open. Now, I know I'll be mentioning this in lab. You can dig and find it within the lecture textbook if you go between chapters, but only males have a defined internal urethral sphincter. There is right at the proximal end of your urethra. Females do not have a defined sphincter in this location. Females do have some scattered smooth muscle fibers wrapping around the urethra that can compress the urethra and sort of act like this internal urethral sphincter. But I'm just putting it out there. The internal urethral sphincter, it's a male thing. So you've got this detrusor muscle contracting, increasing pressure. The relaxation of the internal urethral sphincter relax. If you have it, it's going to open. However, you have your external urethral sphincter as well. And the external urethral sphincter is made out of skeletal muscle. And that means it's under voluntary control. So if you do have the increase in fluid pressure within the bladder, the internal urethral sphincter relaxes, when that urge to urinate begins, you can consciously constrict that external urethral sphincter and prevent urination at that time. So you do have the ability to regulate voiding of urine. However, if your bladder fills to about 500 milliliters of urine, the amount of hydrostatic pressure actually created within the bladder itself is usually high enough that it will force your external urethral sphincter to open anyway and trigger urination, which is why you can deny your micturition reflex for a while, but at some point you're going to get so much pressure that you will urinate whether or not you want to. And so you can deny this reflex for a while, but not indefinitely.